This figure, this diagram is your friend. You want to be able to like reproduce this. So even if you think your drawing skills are terrible, you can draw a bean shape with waves in the middle.
It looks like a brain. I thought it was a brain.
You think it looks like a brain?
Yeah.
Sure. Okay, the folds in the middle. So the mitochondria, we talked very, very, very briefly about the structure. Here's the structure of a mitochondria. Okay, so it's got two membranes. So the outer membrane and the inner membrane. The inner membrane is what is in folds. Okay, so there's two spaces in the mitochondria that you need to know about. The innermost space on the inside is this lighter color, and it's called the matrix, like the movie Matrix. Sometimes it's called the mitochondrial matrix, but it's the very inside of the mitochondria. So if you slice it open, it's going to be the innermost layer. And then the space in between the two membranes is called the inner membrane space. So it's not very hard to remember what they are, but it's still something you need to be familiar with for the terminology's sake. So again, we have the outer membrane, the inner membrane space, then the inner membrane, and the matrix is on the inside. Okay? Those are going to be important because different parts of cellular respiration occur in different places in the mitochondria. Except the very first step, that doesn't even take place in the mitochondria. So that takes place in the cytoplasm. So prokaryotes and eukaryotes can both perform the first step of cellular respiration the same way because they both have cytoplasm. Prokaryotes just can't do the other two steps the same way because they don't have mitochondria. Okay, so we've got the three steps. There's glycolysis. Okay, there's pyruvate oxidation and the citric acid cycle, which again is called Krebs. Then there's oxidative phosphorylation, which has two steps, two parts to it. So there's the electron transport chain, which is ETC, and chemiosynthesis. And I'm sorry, I wrote that wrong. Chemiosmosis, not chemiosynthesis. Okay, chemiosmosis. So where's glycolysis happen? Right here. Where right here?
Where's the stage?
Okay. So stage one, where is this? Where is this happening? Outside of the mitochondria. So it's in the cytoplasm. Sometimes it's called the cytosol. So the difference between cytoplasm and cytosol is the cytosol is just literally like the liquid. It's not the organelles. The cytoplasm is technically the liquid plus all the organelles. Okay, so that's the difference between the two. So glycolysis happens in the cytoplasm. Pyruvate oxidation and the citric acid cycle take place in the matrix, so it's in the innermost portion of the mitochondria. In order for that to happen, pyruvate has to be pulled into the mitochondria, so that takes some energy. The third stage happens on the inner membrane, so it's in between the inner membrane space and the matrix, and the molecules that help make that possible are actually embedded into the membrane. So it's kind of like talking about proteins being in the membrane and the fluid mosaic model of a cell membrane. The inner membrane of the mitochondria has proteins embedded in it that help make oxidative phosphorylation possible. So we'll talk about what those proteins are and how that works, okay? So the three stages. Turn and tell somebody the three stages. I'll cover it so you can't see it on the board. Good. Citric acid cycle or Krebs cycle, good. Okay, oxidative phosphorylation. Okay, so let's break just that term down so that it makes a little more sense. Maybe that'll help you remember it. In this case, oxidative does not mean losing electrons. It actually refers to oxygen this time. So oxidative phosphorylation is possible because of oxygen, and phosphorylation means adding phosphate. So if you phosphorylate something, you add phosphate to it. Okay? All right. So guess which step we're talking about first? Glycolysis. Step one, right? That totally makes sense. So what you need to know from this section. Glycolysis means splitting sugar. So the gly means sugar, comes from glucose. Lyse means to split. So you're literally splitting sugar when glycolysis takes place, which makes sense because we start with a 6-carbon sugar, and then that's broken down into 2 pieces, and they're both 3-carbon pieces. That's the pyruvate. Okay. So when glycolysis occurs, we oxidize glucose. We take electrons and hydrogen atoms away from glucose to make pyruvate. So this figure shows the overall reaction for glycolysis. Again, glycolysis is more than just one reaction. So this is similar to the overall reaction of cellular respiration. It just shows the overview and not all the pieces. So you would need to know that with glucose, we have 6 carbons, so each ball is a carbon. 2 ADP and 2 phosphate are used to make 2 ADP. It's one of your net results. 2 NAD plus are reduced, so they have electrons added to them to make NADH, and there are some hydrogen ions that float around from that too, and we end up with 2 pyruvate. Okay, so these ATP are made through a process that's called substrate phosphorylation, and that's what's in figure 6.7b, so that's this one. And substrates, I don't know if you remember learning about enzymes before, but enzymes tend to work on substrates. So enzymes are substrate specific. They can only fit one type of thing in them, which is why if an enzyme's formed the wrong way, it won't work anymore because the shape doesn't match. So these enzymes have a spot for ADP and a spot for phosphate, and they add the phosphate to the ADP. Okay, this is a slower process than oxidative phosphorylation. So there's substrate level phosphorylation and oxidative phosphorylation. Glycolysis has substrate level phosphorylation. Okay, so you're using an enzyme to add phosphate to make ATP. Okay, so from figure 6.7a, you should know it's an overall reaction, so you want to know the reactants and products. And then you should be able to explain this figure and talk about what substrate level phosphorylation is. Okay. This page, oh my goodness, you don't have to know most of it. Pretty much all you need to know is glycolysis is 9 to 10 steps. Okay, this shows 9. I've seen a couple of versions in textbooks that show 10. So it's quite a few steps. You don't have to know any of the intermediates. You should know we start with glucose, we end with pyruvate. Okay, from one glucose molecule, how many pyruvate do you get? 2. So we go from 6 carbons to 2 3-carbon chains. Okay.
I just saw 3. Okay, I saw 3.
That's okay. So a lot of people or textbooks or teachers will break glycolysis down into steps where you have to put energy in and then steps where you get the energy from. So some of you pointed this out when we were talking yesterday. We do spend ATP for glycolysis to take place. How many ATP get spent? 2. How many ATP do we get? 4. So that's why back on this page, the overall reaction shows that we get 2. Okay, your net gain is 2. Hopefully that's how things work. If you invest money, hopefully you get money back. That's kind of the point of investing. So you should know that you spend 2 ATP, but we get 4. And again, this takes place in the cytoplasm. Coolio? Okay. Now we're talking about the next step, and this is a 2-parter. So there's oxidation of pyruvate, and then there's the citric acid or the Krebs cycle. Okay. So the first part is pyruvate oxidation, and that's shown here. So there's 3 parts to it. Carbon dioxide is removed, and an NAD plus becomes NADH. And then coenzyme A, which is actually made from a B vitamin, again, B vitamins, they're kind of helpful, is modified, so it's called coenzyme A once it's changed, and it's added to acetate to form acetyl-CoA. So we go from pyruvate, which is 3 carbons, we lose a carbon, so then we end up with this 2-carbon molecule. Okay. So every time reactions take place, or every time we have steps of cellular respiration, we're oxidizing the molecule we started with and removing energy from it and adding it to electron carriers, or we're making ATP immediately. So in glycolysis, the electron carrier that was in glycolysis was NAD+. So then it became NADH. Now NADH has a couple of extra electrons and an extra hydrogen ion. In this step, we get NAD plus becomes NADH, so it has a couple of extra electrons and an extra hydrogen ion. It's going to carry those until the 3rd stage of cellular respiration. Okay. So the only way that this can take place is if the pyruvate is transferred into the middle of the mitochondria. So this doesn't happen outside of the mitochondria, it only happens in the matrix. Okay? With me so far? Okay, I don't see anybody sleeping yet, so that's good. Okay, so the first part was pyruvate oxidation. The second part is this citric acid cycle. So that's shown here. This is a simpler version than what's on this page. Okay, this page shows all of the intermediates. So it shows all of the names of everything. You don't, again, have to know those. Okay, so you just need to know the overall big stuff. So in the citric acid cycle, sometimes called the Krebs cycle, acetyl-CoA, so this enzyme comes in, or sorry, this product comes in and is used to make citrate. Okay, and I'm going to switch to this side just so you can see the notes I wrote too. So the 2 carbons are joined with this 4-carbon molecule, so we have 6 carbons. That's called citrate. So that's why it's called the citric acid cycle, because citrate in water makes citric acid. Okay, so we have the citric acid. It's oxidized, so you have NADH formed again, and CO2 is pulled off. Okay, so we go from 6 carbons to 5 carbons. Then our 5-carbon molecule becomes a 4-carbon molecule because more carbon dioxide is pulled off. You get NAD plus to NADH, and at this point we make another ATP. What kind of phosphorylation is this production of ATP? Substrate level or oxidative? What does oxidative phosphorylation need? Remember? Oxygen. Okay. There's no oxygen here. So this is substrate level phosphorylation again. So glycolysis had substrate level phosphorylation. The citric acid cycle has substrate level phosphorylation. The third step of cellular respiration is called oxidative phosphorylation. So that's where the oxygen's used. Okay. So again, citric acid cycle, we get 2 carbons that come to make a 6 carbon. Carbon dioxide's pulled off, so then we have 5 carbons. Carbon dioxide's pulled off again, we have 4 carbons. And then these molecules are oxidized some more to finish off the cycle. And another electron carrier is FAD. So FAD picks up electrons and hydrogens to become FADH2. And then here NAD plus becomes NADH again. So in one turn of the cycle, just looking at this picture, how many NADH do you get? 3. Okay, so it happens 1, 2, 3 times. How many FADH2 do you get? 1. How many ATP do you get? Also 1. Okay, this is the kind of stuff you should be able to answer looking at a diagram like this. Can you interpret how many of things you get? Can you interpret how much goes in? So think back to glycolysis. What's the product of glycolysis? Glucose is a reactant. It goes in. We get pyruvate from glycolysis. How many pyruvate molecules do you get from glycolysis? 2. So every time this happens, for one glucose molecule, it's happening 2 times. Okay, that's just something to think about. Questions about this? Okay, it's lots of words. You don't have to know all of these. That's helpful, hopefully. But what you do need to know is summarized here, and it's the stuff that I mentioned. Okay. All right, so we'll move on to the last step. This one seems more complicated than it needs to. Okay. So oxidative phosphorylation is 2 steps. There's the electron transport chain, which is this part, and then there's chemiosmosis, which is this part. Okay. So the electron transport chain does exactly what it says it does. It moves electrons. So what was carrying the electrons?
NAD plus.
So the NAD plus had electrons and hydrogen added to it. So NADH is carrying electrons, and FADH2 is also carrying electrons. So this is where these molecules get recycled, because at the electron transport chain, they drop off the electrons and they drop off hydrogen atoms. The hydrogen atoms are pumped across using the energy of the electrons. So the electrons get moved from molecule to molecule, and then they're pulled back out. Okay, the electrons end up with oxygen. The hydrogen atoms that get taken off are pumped through, so then we have a lot of hydrogen ions here and not as many here. So that forms a gradient. Okay, higher concentration, lower concentration. So that allows diffusion or osmosis of these hydrogen ions to take place. Okay, so the NADH and the FADH2 donate electrons and hydrogen. The electrons are pulled through the chain and they join with oxygen. They'll make water. And the hydrogen ions are pumped across to make a gradient. So high concentration, low concentration. So then at that point, if there's a pathway for the hydrogen ions to travel through, which direction are they going to travel? This way or this way? Down. Yeah, from high to low concentration. So that pathway is this enzyme here. It's called ATP synthase. What do you think ATP synthase does?
Synthesizes ATP.
Perfect. Okay, I'm trying to walk you through it. It's really not that complicated. It just is a lot of words all at once. Okay, so ATP synthase literally just makes ATP. So it's a motor. It's with pieces that look a lot like a motor. There's an axle, there's a shaft, there's a part that spins and rotates. And because of that rotation, it makes ATP. So it allows the hydrogen ions to come through. So the hydrogen ions power the motor turning, like fuel powers your engine. Okay, and then the moving of the rotor of the rod is what makes the ATP. So this is oxidative phosphorylation. What's happening right here? Substrate level phosphorylation happens in the first two steps. This is the oxidative phosphorylation. And this is where cellular respiration makes most of its ATP. So this is where you get big bang for your buck. Okay? This is the best return on investment that you can get. Okay? So what you need to know from that? The electron transport chain is where the NADH and FADH2 donate electrons. And at that point, hydrogen ions are pumped across the membrane. O2 is what pulls the electrons through the chain. So if there's no oxygen, this can't happen. Okay. The hydrogen ions form a concentration gradient, and that powers the ATP synthase. So again, the two parts of oxidative phosphorylation are this chain, and then the chemiosmosis. Are there any questions I can answer for you? I'm sure you'll think of something at 3 a.m.